Hello, this is group B27 and we will be discussing the importance of vestigial tissues and traits. Firstly, the question at hand is why do organisms hold on to vestigial traits when there's no real function? Next, this has been a hot button issue ever since Ernest Haeckel in 1866 first coined the term ontogeny recapitulates phylogeny, which means basically just that an organism goes through steps that resemble the evolution of its ancestors. Previous theories for recapitulation include terminal addition and condensation, but there was a lack of evidence, so that fell through with Lamarckism. The current theories are the complexity correlation effect, which basically just states that simpler, more ancestral traits are the building blocks for later, more complex mutations. Therefore, there's still a purpose, although there's no function. And developmental disruption force basically states that there's a tendency among organisms to avoid anything that may alter a successful phylogeny.
And now we're going to look to see the data and results of various experiments and studies to see how this addresses the question at hand. So firstly, we're going to look to ontogeny recapitulates phylogeny, and that's addressed by the first graph on the top right, which has development order on the x-axis compared to evolution order on the y-axis. And essentially, development order is how an individual organism develops over its lifetime in comparison to the evolution order of the whole species. So we can see that certain traits that are developed early in the life of an organism, or actually those traits were made early in the evolution of that species itself. So, and we see a positive correlation. And now we can look to the bottom right hand and we see this graph over here. The ontology recapitulates phylogeny, and the mean score looks to see the correlation between the two, and at this we can see that it's 0.27. The CC, which indicates the complexity of the traits, indicating how this interacts with the ORP. Less complex traits are made first before more complex traits. The last column is the differential between these two columns to see what is the difference in the impact of the CC in comparison to the ORP. So next, we have an experiment that looks to flowers. And in flowers, we look to see the staminoid, which contains pollen that is sterile, so it can't reproduce and produce other flowers. And this experiment looks to see how whether, even if you have a staminoid which has a sterile pollen, whether it still attracts organisms such as bees to visit and try to obtain the pollen. And we see that the two that do have sterile pollen in comparison to the two that do not, that they all have a negative downward slope, with additional reinforcement of that evidence with the bottom evidence stated here, which still shows the number of visits for unmanipulated plants, the control plants, and the staminoid. And they all have around 140, 150.
Next, we're going to be looking at the fluctuations in vestigial and functional traits within the soldier and disperser morphs. So as you can see, the dispersers' wing lengths are not vestigial, they are functional, so these disperser morphs can actually fly while the soldiers cannot. They are vestigial wings. And as you can see, the soldiers' average wing length is much lower than the dispersals. However, the standard deviation of the soldiers' wings is actually much higher. So there's greater variability within the soldier's wings than the disperser's wings.
So now we're going to discuss the implications of the data results that we just saw. So first, we looked at the ontogeny recapitulates phylogeny and timeline. So we saw in the earlier graph that there is a linear progression in an individual's own growth in comparison to the species, which essentially means that when an organism is growing from its first stages to later in life, that the parts of its body are developed in the same way of the evolution of that organism. Secondly, we're going to look to the complexity correlation, which indicates that certain structures are used for building blocks for evolutionary traits later. So, as we saw with the evidence of the flowers in that experiment, that regardless of whether they had a stomata or a stroman, which didn't actually have this stroman, was a building block in order of terms of using for other secondary functions, such as bringing host animals into for pollination, and also as well as using that pollen as bursts to stop some organisms from coming to the plant, which indicates that these simple structures were still helpful in order for the evolution and for larger functions and other secondary functions that are later to become primary functions. And we also see that those functions are carried down throughout the generations, which indicates that when in a timeline, in a short span of time, tissue becomes vestigial and it learns a new action, that action can be implanted and can be behavioral and can be transmitted down in the generations.
The increased variability within the soldier morphs hints towards the possibility of mutation. This is shown through the increased standard deviation, showing increased variability. This is a great example of developmental disruption force, because although the wings are vestigial on the soldiers, they still exist, and they still have them to hopefully one day mutate into a useful organ.
So now we're down to the conclusion and about the take-home message. What this basically indicates is that these vestigial traits, even though that they might not have their original functionality being used, that they're still influential in the day-to-day lives of these individuals, or how for evolution. When we look to see the complex correlation effect, it states that in order for to have any evolution, we still first need to have that organ in place, which indicates that regardless of whether the tissue does actually function or does not, that it still has some indication of helping to help these organisms to evolve.